to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 78th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for over 30 years. I'm here with my co-host Dominic Guarino.
Hi, my name is Dominic Guarino and I've been in the bioanalytical LBA, biomarker, flow cytometry, ELISpot, you name it, space for about the last 30 years.
Dom and I are members of the growing scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. We're thrilled to have you listening to the 78th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on the topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're ecstatic to have you here, and we're looking forward to a fun episode today. To kick us off for podcast number 78, Dominic is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, so the main portion, we're going to turn our focus to our global services here at KCAS Bio. We'll give you an update on the latest news from Lyon and the rollout of our collaboration with Crux Labs. And most notable was my trip to Lyon that we'll cover here. And we'll give you all the lowdown of all the exciting strategy and growth of our Lyon site, our sister site there in Lyon. And also, as I mentioned, our collaboration with the Australian group down in called Crux Laboratories. So exciting topic, John. I can't wait to dive into it, but yeah, should be fun. All right, John, let's just jump right into the news. What we got? What's up for our first article?
So it's that old chestnut. We're back on antibiotics. So this article is new antibiotic kills off deadly bugs but spares the good ones. And this was basically a research group at the University of Illinois Urbana-Champaign presented the data demonstrating that their antibiotic lolomycin could kill deadly bacterial strains in cells in mice without affecting beneficial gram-negative strains that colonize the gut. So this is what I say. It's an old chestnut. It's a little bit tongue-in-cheek, but we keep coming back to it because obviously bacterial resistance is a big deal for us now, and even more so in the future. So this team had a series of old drugs developed by AstraZeneca. They tested them against gram-negative bacteria. That they showed not much effect, but I think it was that they didn't harm the gut, which was a signpost of a way forward. So they actually tweaked the compound, obviously changed its structure around, and it actually works by disrupting the activity of the LOL system, which is a bit of a laugh in it. Transport system exclusive to gram-negative bacteria that moves lipoproteins from one part of the microbe to another. So the one candidate they came up with is lolomycin. And in a series of tests, they found that it could. So they basically looked at it. I think it's in before they went into mice, it could kill off 90% of E. coli, K. pneumoniae, and E. cloacae that had been taken from patient samples that had been no effect against either gram-positive bacteria or gram-negative bacteria that populate the human gut. They then tested in mice with sepsis and pneumonia, found that it could it really prolonged survival of the mice compared to placebo, and then in comparisons with amoxicillin and clindamycin showed again good effect, but didn't have the bit where amoxicillin and clindamycin would impact the good bacteria. Lolomycin largely left them alone. So it's basically indicative of a potential path forward in terms of a new way to approach antibiotics.
Yeah, no, I mean it's John, it's really cool science. I think they kind of indicate they're still a little bit away from maybe having this effective, but this is really a breakthrough thing. and maybe I'll just rehash some of what you said, right? Like we got all these symbiotic bacteria. I think of them. You need gut bacteria. It's kind of don't look in your eyelashes too much. There's bugs in there, John. Right? We're just a host for a whole bunch of stuff. Right? Don't want to freak anybody out. But like, and I haven't taken an antibiotic in a long time, but it tears you up a little bit, right, John? And this, they're saying here, this would be able to, it really could be a couple things, right? You could give a massive whopping dose of it if it doesn't hurt your, I call it endogenous bacteria, your gut bacteria, whatever you want to think of it. Because this would be a treatment for sepsis and all sorts of, any sort of major bacterial infection. You could now give probably a higher dose, right? A more frequent dose. Although they're still worried about resistance with it, but this is a great engineering, great science. They found a way to just kill the bad ones, spare the good ones, right?
I think it's interesting to say, it could be years before this or drugs like it are ready for obviously for prime time. They need to find that. It is good that they're thinking about risk of resistance, because obviously that's something you have to be thinking straight off. But then how do you combine it with other antibiotics? But also it's like, but who do we partner with? Because this is a research group, and when you've got large pharma aren't really showing much focus in this area, who's going to pick it up and run with it? Because as we've talked about, even those antibiotics that have made it to the market or been approved in recent years, the companies that have produced them have made no money, and it's a really strange conundrum that we need these antibiotics, but the emphasis is not there in terms of research and development and getting them to market.
Yeah, there's a couple of things. One, that finally the market itself, right? It's not one of our articles or anything, but there's a massive boom in IPOs, biotech dollars. So some of it may be going towards this, John. We're turning a corner. If you've read some recent articles, clearly the environment's improving very, very nicely for biotech specifically. And I think these are in that biotech, although pharma could be involved. But I think you will see more money go outside of everything's focused on immuno-oncology, right? There's a lot of autoimmune, or we'll call them immunological disorders, right? Rare diseases. That's where all the money's going. But I do think it will shift towards this. And then, the other thing that's happening is they say that there's a record number of infections, whether it's flu, whatever it might be, no standard cold, even COVID a little bit, right? But that's because everybody's out being active again, right? So that, and I think as we start to, because I do think this is one of the first true summers where everybody's kind of out, and clearly if you've been in an airport, everything's upticking, right? It's only natural that you're going to see 5-year, 6-year highs in infections, right? We're all out together. It's not alarming, but when it starts to bring this to the forefront, John, that's where I'm going. It'll start I think that's what I mean. We'll start. You're right. Right now, we haven't seen a lot of, we're not that we see everything at KCAS Bio, but we do, right, John? We see a lot of the drugs. And we haven't seen any in this space, but it's great to read this article. And I think there'll be that shift in the coming years as the market, just in general, as money kind of loosens up, right? Because the money never went away. I think it just got tight.
Sure, and you're right. As the need becomes more evident, yes, people will move towards
it.
But it's also we're moving towards the need when it presents itself, rather than anticipating the need and trying to get ahead of it, which is a bit of an issue.
Agreed. It's better that's a reactive state, John.
Yeah, sure.
That's the worst place. Because it didn't go, they did make some breakthroughs with MERS resistant, right? MERS. There is some antibiotics that kind of helped with that. But you're right. And there's still I think as you see more and more people in general, they stopped. A lot of surgeries were pushed back. We're still getting caught up in some of that. And I'm not knocking our hospital system, but there's still a major problem there with infections in hospitals, and this is where it has probably the most application.
I think we're all still playing catch up for that 2-year period where we didn't do anything. I mean, I'm planning a trip back to the UK this summer, and it's catching up on things that I didn't do in 2020 and 2021 where I couldn't. Yeah. And it's sort of getting back in touch with family. It's going to see friends in other parts of the country that would have been part of my normal plan, but everything shut down for us all personally, and so you are playing that bit of catch up.
Everybody was weddings, things like that. Everything just kind of see now we're getting back into more normalcy, and as you said, there's just this normal. You're going to go back to that normal kind of travel and living, and I think we will see upticks in infections. But I don't think it's anything alarming because we have good drugs like this that are in the pipeline.
One point I wanted to make about this, it's interesting when we talk about when we talk about antibiotics, you're right, there are things coming through, but it's always talking about a single drug that is doing this, and it's always like isolated. When we talk about some of these other infectious therapeutic areas, you're talking about multiple companies working on something at the same time. So that's the difference in the amount of effort. It's always like one at a time with the antibiotics, and we'll keep revisiting it because it's obviously really important to us as a human race. But it's still not there where there's this concerted effort in terms of developing antibiotics that are going to help us in the long term. Anyway,
well said, John. Amen.
On that grumpy note, let's move forward. So this is this is another thing for endpoints. Headline was "Invisible Hand of Inherited Genetics Can Drive Cancer Risk," new study suggests. And so this is talking about how genetic mutations can drive the growth of cancers, and for a long time, it's thought that those mutations are acquired over the course of a person's life. But there's been a recent study that shows, for breast cancers at least, that actually some small genetic variations present at birth can actually shape the aggressiveness of a disease that emerges decades later. So it basically it can either block the tumor's growth or figure out how the tumor is going to grow. It can help or hinder the immune system, and also help guide how it will react to treatment, etc. And this is from a group at Stanford University. So they looked at genomes of nearly 6,000 breast cancer patients, and they through modelling, they came up with a genetic risk score to determine how likely cancer is to escape the immune system's natural defences and progress to more advanced disease. And so this is like, again, this is early stuff, but this is more looking long term to how it might help diagnostics in terms of predicting for people, and then obviously downstream treatment if cancers emerge, etc.
Yeah, this is a great article, John, right? So there's some words in here that are new to me, and that doesn't happen all that often, but these they call them germline-derived epitopes, or GEB.
Sure,
right?
Yeah.
And, epitope has a big meaning to it, but they kind of view it as molecular signposts. So this does have this great diagnostic potential to it. So instead of like looking for the needle in a sea of they even talk about traditional liquid biopsies and how you kind of try to key in on it. This is, if I'm understanding this, they're doing massive, gene screening and looking for these GEBs, right? That's kind of what's happening.
Yeah.
And so
if I could just step in, they just say that, like you mentioned, they're basically looking for epitopes that cells throw up to help the immune system differentiate the body's own cells from infected or cancerous ones. Those more subtle clues actually help them figure out how genes impact the course of cancer rather than its occurrence in the first place. So I'll throw it back to you.
Very interesting, though, John. So you hear me talk, epitope is a word, like antibodies bind to certain epitopes on cells, right? There's epitopes on the CD3 receptor. I could go on and on about epitopes. Discontinuous, continuous, meaning the way a protein folds. Sometimes you might be binding to an epitope that's amino acid 99 and amino acid 500 because of the way it's folding, right? So that's an important immunological concept there about discontinuous, continuous epitope. But they're talking about, they don't know what the epitopes are yet, which is fascinating. I love science, John. I love it. They're so early on. They're like, man, we're on to something.
Yeah.
It could be a diagnostic screen, but man, the pharma world, the vaccine worlds, right? They're going to love this stuff. If they could start creating epitopes that you could start trying to target as being, and John, it's so early. They don't know if it's a diagnostic tool, right? They don't know if it's predictive, but it's on. I think this, and as I read more of this, they're really onto something, right? It's in like the milk ducts of breast cancer. I mean, this is a really nice advancement.
So if I could just
describe, go ahead. I'm loving this.
Yeah. So they basically, the lead scientist. I've just dropped my paper on the floor. Hold on a minute. The lead scientist is a Christina Curtis, who's a director of Artificial Intelligence and Cancer Genomics at the Stanford Cancer Institute. So she basically devised a metric to quantify the genetic variation that she called the germline-derived epitope burden, or GEB, in pre-invasive breast cancers that hadn't spread beyond the milk ducts. The researchers found that a high GEB score was linked to better outcomes because the cancer cells were more likely to have signaled to immune cells that something was awry. So then she says the immune system can seemingly see those nascent or early tumor cells and eradicate them. We never even see a cancer emerge. So this is the invisible hand that helps.
Very powerful.
But the immune system doesn't always win the fight. Some tumor cells can escape. If they start spreading, the same genetic diversity that was once protected becomes a liability. In an invasive tumor, which has developed ways to evade the immune system, a high GEB score is linked to immune suppression and poorer prognosis. So it's really interesting that it's that counterbalance piece, It's like, if your body detects it, you're good. If your body doesn't quite catch it, then it really isn't good, and so it's really interesting.
You're getting into tolerance, John, right? So if you can see these GEBs, you're all set. But the moment the body tolerizes them, or somehow the mutations occur where they're no longer signals, invisible hands for cells, because they're kind of pre-tumorous, right? These GEBs come up, the immune system's killing them. But as soon as it evades it, that is this concept of break intolerance, which you'd win a Nobel Prize if you could describe that, right? That means that they start to become ubiquitous, and now they're like mock, right? They're like viruses do. Viruses do this as well. Now they're there, and the immune system's not seeing them, and then they proliferate, right? As soon as they evade the immune system, they take over like a vine, like a weed, like a deadly weed in your garden, John. That's what this becomes. Right, that dual edge that you're talking about. So initially, these scores are good. You want these things, but as soon as they don't see them, it becomes like that dandelions on your lawn. So good stuff, John. Great. I don't know what else you want. I do. It's a really cool article, though.
I do like the final quote. It says, "Curtis has filed a patent related to the work on germline genetics. When asked if there were plans to translate the diagnostic or therapeutic applications into a biotech company, Curtis declined to answer." I can't really comment on that right now, but really excited about the implications of this. So watch this space, because there'll be something coming, I'm sure.
If I was an investor, I'd be interested in talking to Curtis.
Exactly, yes, indeed. Anyway, let's move on. This is actually, I pulled a number of things into this because radio pharmaceuticals are a hot button topic.
I can't believe it, John. Yeah, it's somewhat surprising, but go on.
It's funny how we never die. We grow up concerned about radioactivity, and I mean, honestly, growing up in the UK in the 70s and 80s, there was always that thing of the threat of nuclear war and radioactivity and stuff, But anyway, obviously things move on. I'm not dwelling on that.
God, I used to hide under my desk. They made us go to the cafeteria.
I remember first time I walked down a street in Savannah, and it had like fallout shelter. I'd never seen that in the UK, and there it was, and just like I can't remember which street it is, but the sign was still there in the 90s.
I just remember looking over at my colleague, and like we're fourth grade, one of my friends, and thinking, if a bomb hits us, how are we protected under this desk right now?
Exactly.
What is this doing?
I'll get a splinter.
What is this going to do? No, but John, radio labels, right? I'm going to say one last thing, right? Remember, radio RIA assays, radio immunoassays, they were supposed to kind of go away. These things were supposed to go. They're still incredibly powerful tools for scientists. Go ahead, tell us about.
So anyway, I'm going to come back to this. So basically, it's been a fairly recent development over the last few years, but radio pharmaceuticals have actually become a hot area in cancer, and it's basically it's effectively like an ADC, but your payload is a radioligand. Two in particular that have been looked at a lot are 225 actinium and 177 lutetium. There's one approved drug on the market. It's actually lutetium based, and it's marketed by Novartis. But you've got Bristol Myers recently bought a radiopharmaceutical company. You've got AstraZeneca are in the process of buying Fusion. There's a lot of interest in this space because it's that figuring out how to deliver, the radionuclide to the spot of action, and then for it to act on the tumor of interest. The reason why we're talking about this today is it's as we've been talking about other therapeutic areas, other kinds of therapeutics. The supply chain has become part of an issue, something of an issue. I mean, you read the multiple articles. It's like so the first one, there was a number of articles. The first one that came out was actually Bristol Myers Squibb have actually paused enrollment of new patients in the phase 3 trial of one of their drugs that they bought through a purchase of Raise Bio. Where they're actually testing it out for neuroendocrine tumors. Part of the issue with these radionuclides is some of them come from Russia, so it's looking out for alternative sourcing. So they expect the trial to restart for fresh patients. They actually emphasised that for currently enrolled patients, they had sufficient supply. But this has downstream impacts that even if this works, you still need to sort out the supply chain if this drug moves to approval. And obviously, with the Novartis drug, you've got proof of concept that these things work. You need to resolve the supply chain now, such that if it does get approved, you can roll it out to patients and treat them. So that was the first one, and then actually it was then emphasised, I think, this week, where there's actually concerns that lutetium-177 may also be starting to see a shortage. In terms of actinium fusion's drugs, that's actinium based. They say they've got their supply chain is sorted, and they think they're good. And then the third article in all this is a company called ITM in Germany, who are actually have made a huge investment on boosting isotope manufacturing to help alleviate this. But I think it's really worth highlighting that we've got this real push that this could be hugely valuable for patients, but the supply chain, if we don't get it right, could really impact our ability to treat people.
Yeah, John, I hope our executive team isn't listening, but if there's an investor out there who would like to start up a laboratory for radio labeling, I think it's a good market right now. No, I'm just kidding. But no, John, all kidding aside, I think this is the supply chain is definitely an issue, but I think if they become I feel like there's a way to do it that's much safer than, say, 10, 20, 30 years ago, right?
Oh, sure. Yeah.
And I do think there's a couple that the Spectron RX, I guess, is a CDMO that can do it, right? I've never heard of them. I think you'll see more labs pop up that can do it. But I don't think it's ever going to go away, the taboo of radio label. And so this supply chain may never really go away. But what I do want to touch on is the power of these, because some of them can be inert, right, John? And then if you kind of throw an X-ray across the body, it can activate them, right? So is there some science in there? I know that's not these articles.
Maybe I'm not aware of that.
I think there is a way to these radio labels. I think there is a way to kind of do a very just like an ADC, like you said. But a lot of ADCs now, they don't get activated until the microenvironment, right? So they've gone to like some sort of advancement in, not only does my antibody not bind unless there's a certain tumor environment, right? My payload will not fall off unless it's in a specific environment, right? So all these kind of advances. I believe I've read about how you could systematically dose someone with a whole bolus of a radio label in it. And if it's in an inert state, meaning until you give it some sort of gamma ray to activate it. So then instead of like having a whole systemic radiation treatment, it's in the body, it's not there, and you can target where the tumor is. Make sense, John? So that, I don't know, maybe I'll try to find some of those future articles, but these radio labels are in the same spaces. To me, they're probably even more of an investment, John. I think I see a pretty big uptick in the.
I mean, it's interesting. You talk about investment, because currently there are around 305 clinical trials involving
Wow, that's a
lutetium
lot.
177 labeled treatments, according to Line. Of those, 155 are ongoing and 35 are active LU 177 programs in development. There are at least 13 phase 3 trials investigating the isotope and 57 in phase 2. Now, there's a couple of things at play here. Both actinium 225 and lutetium 177 are rare isotopes to begin with. So if you've got that's why you need the demand. That's why you need the manufacturing. I think the attraction of them is they've got short half lives, so you administer them, and then it's a relatively short time with the half life
pretty inert
decay
too,
that
right?
they're inert, that they are they're just in the background of your body. So that's why these two isotopes have become obviously the two favored ones
They're
out there.
pretty harmless too. Like they can't penetrate paper, right? I think they're like.
I'm not sure. Actually, that's an interesting question, because I'm not sure what kind of
I
radiation
know what
both are.
actinium is. I don't know the other one that much.
I would suspect you're probably right. That's another reason for the choices. Yeah, I mean,
I
yeah.
think we have
Again, remember
licenses
that
for
in
both.
in,
Sorry.
in high school classes where you got the alpha radiation is stopped by a piece of paper, beta is stopped by something else, and then you got the gamma
Yeah.
radiation, and you need lead, and don't go near it. And so yeah, these will likely be alpha emitters, but
Yeah, I was at the dentist and they put that lead thing on and took my. I'm like, oh, I still don't love these things, but no, you're. I know actinium is for sure. I think we have licenses for both, and I don't know if you have more about this
well,
article. But
I think it's a case of you because they've got short half lives. You don't necessarily, well, for us as a bioanalytical, we don't need licenses for both, because they decay, this samples decay quickly enough that by the time they can be held and then shipped to the bioanalytical lab, such that the radiation is background and they're considered cold.
we recently redid our license so that we could accept those cold samples.
Okay.
But
Okay.
yeah, I
Sure.
think even though they are a nerd, I think look,
Okay.
I don't know the entire regulations, but we are licensed for actinium. I don't
Oh
know.
yeah, absolutely, we are. I'm not saying. I know there's a process you can go through to make sure that the samples
Well,
are
I mean, to your point, John, the
considered
reason
cold.
why we brought it back was because there's more research in this space. We
Sure.
were an RIA lab for decades, and then when we moved into our new building, we said, "Well, there's no demand for this." And then here we are, 2 years later, carving out space so that we could put in a radio lab, a little bit of real estate for um, radio labeled or radiation, right? So
Yeah. So
we're
as we
back
can.
to doing it. But I want to be clear, we don't test samples
No,
that, right? Yeah, we don't do RIA assays. This is solely so we can receive cold samples and do non radio assays, right? An
or
LCMS.
to clarify, yeah, it's for like some things like studies, or
Yeah.
we got carbon 14 administered, so it's there as a small proportion of the total carbon in the sample, but it's basically there's other parts of the study being used to determine through radioactivity where it's going, but we're measuring off the coal, but there's low levels of C14 in it. We know ahead of time that's above background, so you need a license to handle that, which we have. So we've got carbon 14, tritium, and a couple of other isotopes which I'm not sure what they are, but yeah, it's there, and it just helps broaden the basis of what you can support. I mean, because if you don't have that license, you can't touch carbon 14 samples. You just can't do it. So it's another box that we've checked to make sure that we've got as wide an array of services as possible.
Yeah, John, I think we beat
we
the
move
radio
on?
label to a dead horse here. We can move on.
Let's move on. Absolutely. I think the supply chain is always interesting.
Oh,
You
yeah.
can't ignore it, particularly as we get into these more and more complex moieties with difficult manufacturing and all that kind of stuff. We'll keep touching on this, I'm sure.
A quick
Anyway,
sidebar. A friend of mine's vehicle is delayed because of the Baltimore Bridge being down. They
Oh,
can't. Yeah, it's been delayed a few months now because that car can't get on land.
yeah,
It's
sure.
interesting.
Yep.
All right. Yeah.
Anyway, let's move on. This is again. I thought this was worth touching on. Basic. Remember back in the days of COVID when we were sitting on here talking. We had no treatments. We weren't going anywhere. We were isolating, etc., etc., etc. But there was the Operation Warp Speed to really push to get something to mark to at least authorized use, so that we could start rolling things out before full approval, etc. So I think that model of very rapidly developing drugs is being looked at, and where else can that be applied? And this is coming from the FDA, and so their thought is they'd like to apply that model to other disease areas, and they've actually selected rare diseases as somewhere they want to go. And as part of that, they've actually selected 7 participants who they are working with to, obviously, how can we work with you to get these drugs to market quicker? And 5 companies have actually made it public that they are part of those 7. And those 5 include Denali Therapeutics, Larimar Therapeutics, Grace Science, Neurogene, and Mortel. So the pilot aficionados support for clinical trials advancing rare disease therapeutics pilot program will bring the early stage participants and relevant FDA staff closer together on communications around clinical study design, fine-tuning the patient population for the sponsors' trials, and more. The program, first announced last September, initially sought to include 6 participants, 3 from the FDA's Centre for Drugs and 3 from its Centre for Biologics. So I mean, I love that we're not just thinking, well, we learned stuff from COVID and not applying it. I love that it's actually, there's some thought being put into this, and hopefully, I thought it was worth raising this because we can watch how these things progress and see if it actually is a successful initiative in other disease areas, because I know that was something that if we could apply what we did for COVID to other stuff, this could be great. And maybe it will be. This is our first test of it.
Yeah, it seems that way, right? You got 7 companies and 6 individuals, 3 from CBER, 3 from CEDAR, and that's in and of itself like moving mountains, right, John, to have that type of collaboration. Before Operation Warp Speed, we had never seen so many companies kind of come together and worry less about IP and dollars and more about public health. And so, this could be a real step up for the rare disease space. Yeah, this is a fantastic collaboration, and it is good to see that we're capitalizing on some of the momentum we gained from the COVID research. And I think this is incredibly promising. And they address some of the key things around patient enrollment and all these little things that, especially like, the pre-existing antibodies in this space is still a little volatile. You know what I'm talking about, John? Like, how much do you invest in screening of patients that have it, don't have it? Do you include them? Do you exclude them? Right? A lot of these, what we're probably finding out is every one of us have AAV antibodies, right? This is something that's been around for, thousands and thousands of years. And so, if I'm a rare disease patient and I have a AAV9 antibody, you're not going to let me in your trial. Well, is that the right thing to do? And I think all of this starts to really hone in on that. And then even the bioanalytical space is still a little bit of the wild, wild west in terms of how you handle PK, right? PD-like, PK-like, right? Those are the terms, because you can't you throw in a bunch of whatever the therapeutic might be, you're not going to be able to produce a PK curve like you would for a biologic or a small molecule, right? That just doesn't happen. More importantly, the drug itself, 9 times out of 10, is not the functional drug product. Right.
Yeah.
So the functional drug product becomes the PK, but we gotta kind of create terms that we can all agree upon, and hopefully these types of groups between CBER, CEDAR, and 7 companies, they can come up with some language that helps identify how we want to talk about this, so that we're not having a lot of confusion, John. Right? You hear people say, "Oh, I need this PK method," and we're like, well, we can't validate a method like that for PK because it's not going to follow the regulations. And it's a circular argument, right? Now it's almost like where the biomarker space was about 15, 20 years ago. We're kind of figuring out how to discern what's qualified and validated there. These therapeutics, whether it's a cell-based gene therapy or just a gene therapy, which is most of what we're talking about here.
Yeah.
The PK on that is still like, I wish we could just create a new word for it. Like, you know what I mean? So that we're not, it should, and it can't be like PK like. Those terms are, do you feel warm and fuzzy when I say like? No, right? PD, that has other meanings to it. So it would be nice if we had like, gene therapy kinetics or something. So we got a new thing. But anyway, that's my little diatribe here, John. This is really great stuff.
Yeah, so I think, like you say, it's mainly gene therapies are what are the ones that have been publicly announced. There's an enzyme replacement therapy with Denali and a protein replacement therapy for Larimar. So yeah, like I say, it will be very interesting to
Yeah,
see.
it's going to cure autoimmune disease too, John. That seems to be a big thing. I know
Yeah,
this is focused on rare disease, but you know, the COVID vaccines themselves have really accelerated the vaccine space for good or bad, right? And for the most part, good, right?
for
And
good.
so,
I'm
yeah.
firmly on the side of good.
Oh yeah, but there's always like anything, a vaccine can have some,
Yeah.
yeah, but definitely for the good. And now we set our sights on the things that are almost holy grail. Like, we don't. I don't know anybody in my family that has any sort of rare disease. I'm blessed like that. But if I did, I'd be dialing these companies, John. Right? This is definitely the cutting edge science, in my
Hmm.
opinion. And more importantly, it's cost effective. These shouldn't be, a CAR T cell therapy. They're great, but that's like a $2 million therapy. If you can make an LNP or some sort of gene therapy, that could be like, $1,000 therapeutic, right? There's a huge implication financially for treating, as well as like the current treatment for rare diseases, enzyme replacement, making a recombinant version or trying to isolate it from patient, healthy donors and stuff. Big expense, multiple injections, multiple hospital visits, right? These technologies, one, maybe two injections at
Yeah,
low
yeah.
cost could cure a lifetime. I mean, John, this is amazing medicine right now. And like I said, I know rare disease of focus, immuno-oncology, but boy, anybody with allergy, immunology, all these sort of autoimmune, arthritis, psoriasis, all these things hold, there's a lot of things to, I don't know if cure is the right word, but to treat those diseases. But these gene therapies could actually potentially cure it, John, right?
Oh
That's
yeah, absolutely. Yeah. I mean,
amazing.
that's certainly where they're pushing on these things. Yep.
Well, that's my 2 cents.
Okay. So shall we move on to the main topic?
Yeah.
So I'm going to give you the stage mostly because I'll roll out the main portion. So we thought so Dominic has been jet setting recently. So the main topic is actually turning our focus to global services. So it's the latest news from Lyon and a rollout of a collaboration with Crux Biolabs, which has been announced, but we'll talk further about it. So while all the rest of us have been slaving in the dark holes of upstate New York and Kansas, Dominic has been exploring the sunny uplands of France and visiting our colleagues in France. So I thought let's kick off with we obviously we had Mark, who was the CEO at the time. We had him on the podcast a long time ago, and it was just a good opportunity to really talk about where have Lyon, how have Lyon moved on since then? Because obviously they're growing. What's the current status of the lab, and we have obviously plans as in terms of working globally between Olathe and Lyon, and I'll pass it back to you.
Yeah, John. So I got the chance to meet our new GM, Musin. He's been there for about 9 months. Very talented individual who's bringing a lot of years of CRO experience to replace Mark. And so I got to spend some time with him, and he's got a vision of how we're going to continue to capitalize. So Lyon, for those of you that don't know it, our Lyon, the KCAS Bio site in Lyon, is their areas of expertise are, they used to be called active biomarkers. So biomarkers is a big part of their services and expertise. Their claim to fame is they had more biomarker instruments than any other CRO in continental Europe, which is cool, meaning platforms like
Mm-hmm. Yep.
MSD, Luminex, ELISA, Simoa, LumaPulse. They had a whole suite of them. And so that suite of services, and they got a lot of expertise in the neurodegenerative space, as well as a whole other bunch of biomarkers, but they really do have depth in neuro biomarkers. And then the other piece they've always had a rich history in is vaccine support by ELISpot and flow cytometry. So in addition to their biomarker ligand binding assay, so they can support a lot of, let's call them soluble cytokines, soluble biomarkers, and they actually did humoral responses there as well. But they also have the cell-mediated responses, and the two platforms for that are flow cytometry and ELISpot. They've got a very rich history of supporting a whole plethora of infectious disease and immuno-oncology vaccines, as well as therapeutics by Flow and ELISpot. This is prior to us ever joining them.
Mm-hmm.
And now they had done some ligand binding assays, John, But since they were acquired by us almost 2 years ago now, it's been about a year and a half. But about I think it's 6 months ago or 9 months ago, they were certified as GLP.
Yep.
So that brought them into the PK and ADA aspect of things. So they've been working over the last 9-10 months with our headquarters here in Olathe. So they've been working diligently to mirror our SOPs. So I can say with conviction that their ligand binding assay team, immunogenicity and PK SOPs are ready to go, and they are working on regulated PK and ADA methods for people. They have one or two projects, one project under their belt, the second one they're working on. But in the discovery space, John, those generic LBA assays, they've done 5-6 of those. So they're really getting brought up to speed. Our headquarters in Olathe we have a 60-person LBA team that's cranking out maybe 75 or 80 validations for PK and similar numbers for ADA annually. And then a whole host of discovery, non-regulated. We got a whole group that's dedicated. We're not dedicated, right, John? We're not siloed, but that focuses on that. And they're turning out, they probably got 6-10 accounts working on that. So all that horsepower is being leveraged into our Lyon team. And they got overall, the Lyon team has about almost 40 head count. They got about 10 of them are kind of more towards the flow space, and 20. And again, there's a lot of swing players in there. I'll call it that, but in more in the LBA and the biomarker space, they do have they got a fair amount of PIs. They have like 6 PIs, and so those teams and then the program management team is very robust. But more importantly, they do have a dedicated development team and then a validation team. So even though the size they're and then John, so that shift happened a few months ago, and I bring that up because that is graduating into being able to increase your capacity.
Yeah,
you
absolutely.
Have that development team, right?
I think a lot of us have gone in that direction where it's like if you split the development off and then have your more production-minded team, it really is. It's a scalable approach.
It is. Doing things. All the hard stuff gets into the development team, and that team is regulated. It's not like it's so they can hold on during the validation if they need be, but if they can transition it into the production team, it's just good camaraderie, and they've got some talented principal investigators there, or principal scientists actually, that has a wealth of regulated experience before joining that team. The team is up to 3 dedicated MD people, which sounds kind of small, but in the same token, the facility, John, man, they moved into that building right before we acquired them. They've repurposed it, and we'll talk about why they're repurposing on the flow end. Give me a minute there. But that lab, they could have easily double the number of people because there's a whole floor below us that's empty. And then they're building a whole building, a new biotech building next door that's under construction. So we have ample room to grow, John.
Oh, nice.
Yeah. It's really good to hear. So we as you know, we chose not to put mass specs in this year. Instead, we put flow cytometers in, and we're going to talk about that in a minute.
Mm-hmm. Yep.
But I assure you, after we get because that team's also getting up to speed with our EQMS system. They did that over a year ago, so their QA is married. It's all lined up with our QA. As I mentioned, the SOPs in the headquarters in Olathe are now mirroring the Lyon site. They've been busy, John. This is exciting news. I was so happy to learn about that. And then Watson Limbs is being installed this week, and we have 3 of our IT people there next week. So Watson Limbs is another huge step.
Yeah,
These are all the signs. Those of you that are listening that have any need for European regulated PK and ADA work for biologics, there's no small molecule there yet. Give us a call. But
we're going to space on the small molecule, though. Yeah.
But that's John. That will come soon after. But part of it is between the Watson Limbs and now they're bringing on 2 new flow cytometers. We're going to talk about that. We don't want to overburden them with new systems and new things all at once.
Yeah.
But they are moving in they were one of the strongest. It still was regulated work. It was GCP work, though, right?
Mm-hmm. Yep.
Now they're moving into that GLP space. They didn't do it historically. They hadn't had a lot of non-clinical or preclinical work. That's changing rapidly. As a 40 plus year organization, we have a lot of our existing accounts that are kind of kicking the tires on them, John. And I'm proud to say that there's no guinea pig moment here. They've validated methods now. They have existing SOPs, and they're ready to rock and roll.
Sure.
Yeah, I mean, sure, they haven't cranked out 60 like we have, but the way we're structured now, and this is really important for those that are interested in our global footprint. When you go to do a project in Lyon, you're going to get all the horsepower of our Olathe site, meaning Shek Kane, our executive director, will be, or some of our associate directors, they'll be there for any sort of operational calls. They'll support them. And then, John, on kickoff calls, they're going to have a presence. You know what I'm saying? So we're going to have a system where, so that we want our clients to understand that whether you're working in Lyon or Olathe for regulated LBA or non-regulated biomarker work or regulated biomarker work, it's one team. I mean that, John. I'm proud to say that. I cannot believe how much work they've been. They've been incredibly busy. I'll take a breath and we could talk about flow, but they're ready. They are ready for prime time, John. They're doing some good things. I challenged some of their scientists about the regs. I mean, you know me, John. I'm gonna interview you, right? I'm gonna interrogate you, give you a dental cleaning, whatever you want to think about it. And I'm so proud to say that Alan was his name. Their chief principal scientist over there. He really knows what they're doing. And more importantly, this is not a new company, John. They know how to turn a crank. Exactly. They've been around for 20 years. They got Christine Baines of the world, Sendrins of the world. These are 20-year experts in this field. So this is not a new shop. I'm just so excited. I'm so happy for them. I can't wait till we start cooking.
And that's the crucial piece, isn't it? I mean, you've got the very strong baseline to build in, but rolling in like systems like Watson is just huge. Fantastic for a lab like that. I mean, I was very lucky in my previous career that we had Watson we had the precursor to Watson from day one, and so the whole place revolved around Watson and the limbs. So we knew how fundamental it was for a lab like any bioanalytical lab to function. It made things so much easier, and it's always interesting to me to hear of these labs that don't have it. And you think, well, how do you do
John, I was here my first 5 or 6 years here. We didn't have it. We had that. Yeah. What was that? DB Lab Cal? It was some sort of custom-made limb system and all that. And I'm not. It just. You can operate without a limb system, but if I want to put 2,000 samples on your doorstep and you don't have a limb system, it's just going to take a long time.
Yes, that's
it. And so the limb system should be installed by they think they'll have it up and running by middle of August timeframe, maybe a little sooner. But like I said, there's 3 boots on the ground over there, and they are, installing hardware and doing all sorts of stuff that's a little bit voodoo to me, but this is amazing. It's just, I'm super excited for them. And plus, having installed Watson limbs at our site 4 years ago now, we got a little bit of a roadmap on how to do it, right?
Exactly.
Everything they're doing over there, and I can switch gears to how they're revamping the lab. Those are all things we do all the time, right? Even in our new building, we've made 2 construction changes, John. You know what I mean? We're just constantly increasing our capacity, and we got such sophisticated systems that tell us, our building's getting about 80% to capacity. We don't let it get over 80%, right, John?
Yeah.
And then what do? We knock a wall down. We do something, right, to increase our capacity. And that's why we're putting 5 new mass specs in there, reconfiguring some freezer rooms, and boom, our capacity is increasing again. So it's just great stuff. And I don't know if you want to ask me any questions. I hope that gives you a good feel to visit. No, I'll say we have a very talented BD team there.
Yep.
And part of what they kind of know it, but they really enjoyed, a 2-hour roundtable discussion from me and a little bit of an ad hoc, but we talked for a couple hours about PK and ADA and just just simple, not simple, but just when do our services pick up, right? So they pick up right in that lead candidate, right, John? We're not a true high throughput company, but you got a handful, 3 to 5. We're at the tail end of the discovery, late stage discovery, the last third of the discovery process. When you've got 4 or 5 candidates, you're making small quantities of them, and you want to do a quick and dirty kind of assessment in vivo in some sort of murine. That's where our services pick up. And then we go straight through all the GLP talks, right?
Sure.
Or, initially a lot of people, smart, good labs, strong labs will do a non-GLP talks and then a GLP talks, right? Kind of all that process there. And then you go into phase one, we support all that. But they just giving them the lowdown on the PK. Obviously, as soon as you get into a GLP toxin on, you need a validated method. But the ADA for biologics is changing, John, as we know. So that's talking about lean validations and educating them on that. There's really not a need to do a 3-tier validation in a non-clinical setting unless you're high risk immunogenicity. And that was the other thing I talked extensively about, immunogenicity risk assessments. Although, we're not a consulting firm, although we probably could, but we can help with identifying, what your needs are, what the regulated requirements are specifically for the immunogenicity assessments. The PK is pretty straightforward, as you know. It just needs to be validated. So I'll take a breath there, John, but it was a wonderful trip. I got to spend about 3 days with the team over there. I'll tell you a little bit about my adventures in Lyon, but any other questions around that? And I hope that kind
No,
of. There is more here.
I've seen the facility, and I mean, it's very impressive. But obviously, I was there as a mass spec person, and to me, everything was boxes, and I didn't really know what any of them did. But it's a really nicely set out.
Yeah. So maybe I'll take a moment to say the strategic growth for Lyon specifically is they've been working for a good solid almost year with our Olathe site to grow the PK and ADA non-clinical and clinical. And we've started doing the non-clinical. So that's the biggest growth in Lyon from my seat on the bus will be that GLP, getting the GLP status, getting some of the existing accounts that are pharma that we have that are French-based buying into that from us, and then some of our existing accounts here with European needs starting to do it. So we've got a handful of projects, and the sky's the limit there. The other thing, they did get the Lumipulse system. They're the first laboratory to bring this system online. It's got the sensitivity of the Simoa, and it's got the same sort of suite of biomarkers, but it's like an immunoanalyzer, John. It's a lot easier to run, should be a lot more affordable. So that's another nice growth area, the biomarker business. And then the biggest growth plan we have is they were redoing the lab. They're going to have 5 flow cytometers there, John, 2 Fortesses, one of the Nova site, which is a legacy instrument, and then 2 spectral flows. And those spectral flows are going to be they're going to be installed, I think midsummer or sometime in the construction should be done middle part of July. The instruments will be installed, and they're hoping to have them running by like Q3, sometime September, October range. They should be on, and when I say that's when we can put projects on it. We're going to do a whole bunch of other PQ, right? On panels, and I can talk more about how we're going to unify panels that we have. We have backbone panels as well as some off-the-shelf methods like TBNK plus panels that are like plug and chug flow, spectral flow panels that we have in Philadelphia. And that Philadelphia site is part of the IQ, OQ, PQ, the PQ portion of the spectral flows at Lyon. We're going to transfer those methods to them. John, the beauty of what I'm about to tell you, we're doing the same thing at Crux. So the Crux Laboratories. Go ahead. Go ahead. I'm on a roll.
Because I think we do want to back up to that bit because we haven't really talked too much about the Crux.
Yeah,
I do want to say. I mean, I've been through this with a European lab and with a US company. I think it's absolutely critical, and I love the fact that we're going in that direction. That you've got that unified front between both your lab in Europe and your lab in the US. You're not isolated from each other. You're thinking.
Yeah, that's like 95% done, John. Sorry, we're about 95% of the way there. We're not 100%, but we will be in another month or so. It's so far advanced.
I understand that, but it's key that you take that approach. I mean, that was something that we did in the past, and it was fundamental to the development of the what really was, for what I want to be better word, the junior organization. But it really helped bring them up, and actually, made them, because we knew what we did here, to help to coach them into what we did, really brought them forward very, very quickly. So I think, and that really helps with customers as well, when you can say that, it doesn't matter which lab you come to, you're still going to get that same same type of service and same level of service, and obviously we're talking between the sites, so we're all aware what each other are doing. That's really critical for client comfort.
Yeah, no, we have plans to maybe send some of our method development scientists there as well to do some training. I mean,
I would
all sorts of
thoroughly
great stuff.
recommend that.
Yeah,
Absolutely, that
yeah, no,
is.
we are. Look, if you sign a contract or do work in Lyon, you're going to get all the horsepower of our Olathe site. That's our sister site. And it's not, John. I speak with conviction because it didn't happen overnight.
No, it doesn't.
They've been working a long time towards this, and I'm so proud of what they've done. I'm so happy that we can now, with anybody who wants to do a GLP validation over in Lyon, they're gonna get all the experience of our Olathe site pretty seamlessly. They've got it worked out, and as I mentioned, we even want to have part of the kickoff calls and discussions so that we don't want our clients delayed or anything like that if we do run into trouble. But that team over there is really strong and poised to do great things and has plenty of room for growth. And so yeah, I don't know if you want to shift gear to
so that's now Olathe and Lyon. So let's move into the global partnership with Crux and how this all plays into the partnership, and if you want to talk through that.
Yeah, sure. So as I was mentioning, we are partners. So to be clear, we have spectral flow cytometers in Philadelphia. We've been doing it for almost a year, I think, and we've got we have a partnership with SciTech, right, John? The people who make the spectral flows that we have. We had an open house with them. We'd like to think we are one of the premier CROs for supporting flow cytometry on the spectral, and that pound for pound, I think we are. You'd be hard pressed to find any competitors that are as focused as we have been on spectral flow over the last few years. Even though we might not have been the first to offer the service, I feel like we've blown past them. And why
can I ask a question?
Sure.
What's the benefits of spectral flow cytometry versus your typical?
Yeah, conventional flow, you're limited to maybe about 20 colors, John, and the spectral flows can go up to like 60. But that's not really the true power of the spectral flow. Conventional flow cytometers, you have to emissions of light through these fluorochromes require compensation on conventional flow. So you may have to comp out like 20-30% of a channel, meaning the red channel and the green channel have overlap. And as you get all into these blended dyes and all these colors on the spectral flow, on the conventional flow, you just got these narrow windows where anything that's in that like gray area gets comped out. You know what I mean? If it's in this, like if it's in, so now you might be only getting like 60% of a given channel. Well, what's in the 40%, right?
Yeah, sure.
So it's not like you're cherry picking, but you were at the limitation of the flow cytometer because it was all noise in those. If you didn't compensate out, you would have, had all these, like, I think of it as leaching or even like resonating fluorescence that you just can't determine what the heck you're looking at. So they call it compensation. So you'd have to. It was a laborious process. There is some automation, some software, but it was a pain in the proverbial butt to have to comp the instrument on a daily setting, compensate it, and then comp your data, and then try to go back into the data and figure out, oh wait, maybe I want this. Can I comp? It had some real downside.
Sure.
So spectral flow, in addition to the increase in colors, there's no compensation. They take the whole spectrum, hence the spectral flow. Every color, you're analyzing the entire spectrum. Now, they do have to unmix, which is because they all kind of, if there's a 20 color panel, you got 20 spectral flow patterns that are all kind of together. You have to unmix them and then kind of assess them. But there's software, great advances in software to help do that, some automation there. Omics is one of the companies that offers it. There's a few others. We're at the cutting edge of that. That's a big part of what separates us from our competition is how we unmix and how you control those experiments, John. That's a big challenge for flow cytometry that we have. It's not intellectual property, John, right? We're not an IP company, but we have some good understanding of it. That's why I'm so bullish on our flow cytometry team. That's a dedicated team in Philly of almost 50 people that all they're doing is flow cytometry. Yeah, sure. Maybe our competitors have labs but not to that degree of like focus and even bandwidth in one location. Most of the labs that have that, they're just turning out TBNK panels and, running routine flow cytometry panels. Where Legacy Flowmetric, that site has been arguably the number one receptor occupancy lab, the number one flow cytometry outsourcing lab, standalone lab until we acquired them. And we've continued that model. So they're not getting up worried about PK. No, they're getting up every day doing flow, John. So that's allowed us to expand our spectral flow. I think we've blown past the competition. And so what are we doing? We mentioned the two. So we're going to, in our Lyon site, we're putting 2 spectral flows there. They should be online, IQ, OQ, PQ, sometime in the September, October range. In parallel, John, and they're busy over there in Philly, they are installing a partnership with Crux Labs in Australia. Now, they are a standalone CRO that we have formally entered into a partnership with. We are installing these instruments, the 2 spectral flows, just like we're doing in Lyon. So we're essentially doing the same thing in Lyon that we're doing down in Australia. Although it doesn't say KCAS Bio on the door, those instruments are being installed by us. We have people going down there to train them, and those instruments will be used for our studies at Crux Laboratories. Now, Crux Labs is maybe like a 40-person shop, 35, I believe. They're not similar size as Lyon, but they do have mass spec and they do have LBA work, John. So okay. We're starting to finalize how that will work.
Yep.
We will be able to leverage their expertise there as well. But predominantly, the partnership as it stands today is focused on spectral flow. And you know why. A lot of the large pharma companies, they need a global footprint. That gives us that European continent, United States, and now over in the APAC region. And we're really going to be I think we're only the second lab that can make that claim to fame, John, maybe a third one. But the one lab that has it, I'll give them credit. They're pretty good. The other lab I'm talking about, they're not so good. They're having some problems. And there are some other labs who have flow cytometry services, but not spectral. Okay.
Sure. Yep.
So we're going to be only the second CRO that has global spectral capabilities.
And so you talk about readiness of Lyon in around did you say Q3?
Yeah, the goal is to have. They're targeting September. But it might be more in that October. As the instruments get installed, We'll start carving out the date, but it'll be September, October timeframe.
And what's the timing for SciTech as part of all this?
Yeah, that's the timing. So the instruments will be installed in
it's the same
July. Yeah, it's going to be July. Both of them are on parallel together.
Okay.
Yep. So we would anticipate both of them going on either same week, a couple weeks apart, but it's really in that late September, early October timeframe. We have to move it up, John. And when I say online, that means they've got a method they validated, and they're ready to go offer services.
Yep, got you. And what will the services be? I mean, as someone who's ignorant in this.
No, it's okay. It's a good question. It's your standard phenotyping, but of course the power of the spectral flow. Our spectral flows are out to about 40 biomarkers right now, or 40 CDs, or 40 markers. They have up to 60. We haven't done that yet, but we have 40 parameter panels that we've worked on. But most of them are not. Most people don't need quite that level, John. Right? Just like anything else, if you generate data, you got to justify it, right? So most of them are in the mid 20s or below, and you do phenotyping, right? Surface staining, intracellular cytokine staining, or any sort of intracellular staining. You could do, standard phosphonuclear, cytosolic, whatever type of proteins that might not be on the surface, in addition to the phenotyping. And then receptor occupancy, John. That's still standard. Sure. We are, it's easy for me to say, we're the best at receptor occupancy, but we're the best at receptor occupancy, John, even on the spectral flow. We know a lot about it. We definitely have invested heavily in that. So those are the major areas there. There's surface staining, intracellular staining, as well as the receptor occupancy. And then there are a spattering of things you can do for support of like CAR T cells and things like that, where we have expertise.
Excellent. So all exciting times ahead.
Yeah, well, I mean, the real excitement, I'll take a slight side note, and Lyon is a wonderful city. It's the third largest city in France. I believe it's about 400,000 or 500,000 total people. Wonderful, John. They got this beautiful church that, I got there like I took a overnight red-eye flight, landed in Paris at 7:30 in the morning, somehow managed to get myself on an 8:30 train, John. So I'm a worldly traveler. Got into Lyon like midday, hadn't slept. And so I went around and looked at Lyon and went up, walked this full mile up, and beautiful, beautiful view of Lyon. And then the next night we're out at dinner, and I don't know. I'm not that into politics in Europe, John, but there was some sort of EU vote that the populists kind of gained control that caused Macron to kind of call an emergency vote. Well, we found out later that there were about 4,000 to 5,000 protesters. So we're out to dinner, and Lyon has a whole bunch of murals. So we're out to dinner in the old part of town, and we have this beautiful French dinner, and we're walking around looking at a couple murals, and I'm there with two of my French colleagues, and then Amy's with me, so the four of us, and we say, hey, why don't we walk over to where your metro stop is, and we'll pick up an Uber over there. So as we're walking, we just hear a bunch of people kind of marching, and we think, is this for the Euros? But it's like 11 o'clock at night. It's like, that's kind of late for a... And sure enough, John, the police start to break it up, and they start moving towards us. And every time we took a click to move away from them, they kept moving towards us. And so now we've got like a bunch of 20-year-olds kind of running away from the police, running by us. Chaos is kind of erupting. And we say, we're not going to walk to the metro. Let's get an Uber. So one of my colleagues orders an Uber, and she's like, it's over there. And I'm like, I don't think we should go over there. We're walking right into the heart of the riot. And she's like, no, it'll be okay. Because apparently Lyon doesn't have this happen to it. It's not like Paris. And I don't want to make it sound like there's riots everywhere, but I don't know. But apparently this is very uncommon. So she's like, oh, we'll be fine. And so, John, we walk literally right into the middle of it. There's like fires going on. And suddenly we hear like fire. I thought it was just fireworks going off. And I see these like firework things kind of streaming down. Well, John, that was tear gas. And so we got now it got a little chaotic for about a 5 to 10 minute clip. I can barely see. Someone finally says, cover your mouth. I'm like, so if you cover your mouth, you're okay. It wasn't like we were in a room that got tear gassed, right? We're like in a city block trying to get away from the tear gas. But John, I can say I've been tear gassed now. And I'm telling you, I could barely see. I was struggling breathing until I put the thing over my mouth, and I'm like, "Oh, dumbass, right? Like you can cover your mouth and be okay." And so there's just kids running. I call them kids, but 20-something, they're running by us. They're running away from the cops. Which one of my French colleagues is trying to get us into the restaurant. The guy won't let us get in. It's kind of getting chaotic because it's just there's never a moment where I truly felt like I was in harm.
So it's very. It just shows how much more of a civilizing influence I am, because I was there a year ago and none of this stuff happened when I was there. It was just,
is it me? I bring chaos.
The worst thing that happened to me was a bit of rain. Well, a lot of rain actually. But yeah, I had obviously found good restaurants, nice places to drink, and had a
I had plenty of that. We still had plenty of that, but there was like a 45-minute. I've never had a policeman point a gun at me until that. I've never been in a riot. I'd never been tear gassed. I'd never been close to arm. I'm telling you, full body armor. I've never had a couple thousand people on the streets running by me, knocking through windows. So. It ends well, but it was crazy. And for me, it was weird because I think my colleagues were getting a little nervous, but I kind of never really did, except for that moment where I'm like, oh my God, I can't really see. And that's frightening. If you can't see, it gets a little, gets a little something,
That can be iffy. Once when I was a kid, I had an accident at home and couldn't see for like half an hour. It's like the worst half hours I've been through.
Yeah.
But in terms of riots, the only time I've ever been an onlooker to a riot was at my high school in Dumbarton. And I can't remember, I think we were shut out on a break and kids went mental, and there's like bins being lit on fire and thrown through the school windows and stuff like that. So, but I was just interested.
Yeah,
I wasn't really part of my scene, but quite fun to watch.
I can send you a little video. Someone did take a little video of it, and you can't quite. You see things burning in the middle of the street, and just chaos around, but not the actual. You can kind of see the smoke in the air, but. Lyon's an incredibly safe city. Paris, I spent another day, and I did take advantage of making the trip over there. So I spent the day in Paris on like Saturday, and it was lovely. The Olympics are this is my second time to Paris. the Olympics are a dual-edged sword because they took and put a bunch of makeshift stadiums, so you couldn't go to a lot of the beautiful parks. It was a little inconvenient getting around. But the Eiffel Tower's got the Olympic rings on it, and we were there at twilight, and it was lit up. So we got some great stuff. It was a special trip.
I'll be honest, I'm a complete Francophile. I've loved every trip I've had to France, and I really like Paris.
I love it.
My wife,
I hit my walking goal every day there. I hit my walking goal. Their meals are so much smaller. I think I do like that lifestyle.
It's so easy to hit walking goals in Europe.
Yeah, it was great. And riding the metro's easy. Even Lyon had a nice little metro system that we kind of took advantage of. The site is in the Lyon's like a kind of a little bit of a biotech of France.
Sure.
It's like their Boston. And so their facility's great. I mean, I can't speak highly enough about the Lyon team, John. But we're a global company, John. I know we're running short on time. Well, not short on time, but I think it's time to wrap up. But I hope that gave you a good.
Yeah, absolutely. I think we were overdue talking about. Well, we were in one way overdue talking about Lyon, but actually the timing is absolutely spot on. So I thought it now was a really good time to talk about what's going on there. So that,
and this isn't my last trip, John. So I think I may be going back over in August or September just to kind of keep reinforcing. And the commercial team has a thirst to learn more too.
So September is an interesting one because I've talked about the chameleons at length throughout this podcast. They're in the midst of a US tour right now, and then they go back to the UK to finish recording their first album in 40 years, something like that, almost. And then they play a couple of festivals, one of which I'm hoping to hit in Manchester. But September, they're playing Lyon with The Wedding Present as their support act. And my wife is like, you should go to that, or we should go to that. And I'm like, yeah, but I've already got a whole load of travel I've got planned. So depending on I mean, we may just do it on a whim, who knows? But yeah, so seeing bands in different countries would be good. Another one they're doing is the Philippines, but I don't think I'm going to.
That would be a better trip, John.
Oh no, but Lyon's fantastic.
I know, but the Philippines sounds even.
Absolutely. It would be great, but then you think the flight involved.
Yeah, you read my mind. That's exactly it. But Taylor Swift, I think, is in Lyon this week. I missed her by a week. I would have gone.
And you know, one of our colleagues went and saw Taylor Swift in Edinburgh.
Oh wow!
Yeah,
I didn't know that.
Hannah Euler with a bunch of her friends went to Edinburgh for a number of days. I think it is cheaper to go and see Taylor Swift in the UK, and we've heard of other people doing the same thing than it was and easier to get tickets.
What an adventure! Our contract. This is Hannah from Contracts. What an adventure!
Yeah.
Former science. She was an analyst. Wow, good for her.
Yeah, I was talking to her on Teams, and she said she was going for this trip. So I asked where and why, and it turned out it was Edinburgh. So, of course, with my connection to Scotland, we were chatting about it.
Oh, awesome!
Yes.
Yeah, no, that's an adventure for sure. Good for her. But no, it was we'll see. Like I mentioned, we're definitely going to make another trip over there and continue to just, make sure I think of it as Lyon is now our sister site, and
sure, absolutely.
All the bandwidth we have over here in Olathe is easily transitioned over there.
Yeah,
and we're even. Like I said, we got some other things planned, and I'll give some other updates around scientists going there, maybe even some methods that we have established in both sites, so that when someone does say, "Hey, how can I get a method in two sites?" we've already got a case study to show.
Yeah, I think it's really important to have people travel regularly between the sites and build up the connection, and it just becomes crucial to the long term.
Well, John, I can't go a podcast without talking about pizza, my green egg, or something cool like that. But I'll just share the one thing that I tried in Lyon that I had never had was squid ink, and it was delicious.
Oh, yeah, you say, what was your verdict?
Loved it. Loved it.
So Venice is big on squid ink stuff.
It was so good. It was like a squid ink soup. I mean, it looked like clay. It looked horrible. Yep. It was so good. Oh my God, was it good?
You cannot diss Lyon food. It's like it's considered the gastronomic capital ahead of Paris.
Yeah, I mean, I did have some duck there, and then we did get a little bit of a Wagovi steak that we shared. So the team took us to some couple nice places out there. It was exquisite, but I never dreamed squid ink soup would be so good. Right? It looked like a face mask, John.
So on that topic, I've talked about doing the winemaker dinners at a local restaurant here called Hazelnut Kitchen, where the guy who's the chef partners with a local winery, or in the first case it was a cider producer. We went to one on May 30th, and the first course was like a bunch of different tasters, but one of them was I think it was, if I remember it was smoked eel on sepia bread, or sepia sourdough, which basically was sourdough with squid ink in the bread, and it was absolutely fantastic. Really, really good. So yeah, I'm a great believer in trying all sorts of foods. But
the other thing was I brought home some macaroons and some other French kind of sweets.
Yep.
And they were off the charts.
Sure.
Did hit the duty free a little bit. Got myself a Glenmorangie 12 and a Talus Dark Special one here, so some Scotch. But overall, and then the other exciting thing is, so I did some shopping and found like the vintage. It's the Matrice area of Paris. So I found like some vintage stores, and man, was my wife going nuts with the photos I was sending. So I got like some Dior pocketbook, Dior purses, and like Dior handbags. Got 2 of those. Got a Givenchy kind of necklace and some Hermès stuff. And she was like intoxicated because you don't find those used things in the U.S. right? That's
no,
and so she couldn't believe it looked like one of the handbags was practically brand new, but it was from like 94, beautiful pink bag. And like, so I did go over into like the real like Chanel store, and you need your passport. And John, I don't carry my passport because I'm weird like that. It's just, I'm like, why carry your passport? You can lose it. So now that I couldn't buy anything there, that's when someone was like, well, you can go to this part of town. And so we went over there and we had a really nice meal in that part of town too, at an Italian restaurant. And the duck there was spectacular. I had some duck. I got some lamb too. It was good stuff. But John, we can wrap up. I don't have much, I'm going to say I don't have much going on this weekend. I don't think, I might have like a round of golf, some couples golf on Saturday, a round of 9 that we're kind of doing this scramble with some friends, and that might be fun. But other than that, I'm going to relax a little bit, and I am going to spark up my big green egg. I don't think I'll be making pizza because my son's working at the pizzeria, Minsky's, but I don't know, John. I'm looking to kind of relax in this week, and I don't have any real great plans going on, and I'm perfectly okay with that right now. Because as you said, I was actually on the road for about 14 nights because I was in Boston visiting family for 6 nights and then flew to Lyon and actually lost 2 nights in there. So went about 13 days without sleeping in my bed. So I'm still, I recovered and all that. And I know I'm babbling on, but my son had his freshman orientation. I get home 11:30 at night or 10:30 at night. I'm back in my house on Sunday night. I got to be in Lawrence, Kansas, about an hour, 45 minutes away at 8 a.m., John. So that was I had been up for 36 hours. I slept a little bit, went to his orientation, and by the way, he's on his way to KU. He's fully enrolled. Excellent. Yeah, how about you? You can take us out. Tell us a little bit about what's going on, and then we can.
I think a quiet weekend here. This has been one of those ridiculously hot weeks, and I think it's always supposed to break this weekend. So our deck is getting closer to being ready for full use, but we're sort of controlling ourselves until it's fully ready before we start using it regularly. But yeah, it's just I think just a quiet, relaxed weekend, to be honest. I'm sort of in planning mode for the UK because the purpose of the visit is to get back together with my sisters after they came out here for my daughter's wedding last year. But the big problem is I can't plan anything until the soccer fixtures are released, and they're not released till July 10th. I want to arrange everything. It's keeping me awake at night because I'm trying to plan the trip in my head, but because I don't know when Hereford are going to play, I can't do anything. So that's going on. That's
you definitely got first world problems there, John.
Right? Oh, I know. It's terrible, isn't it? Yeah, but it's like I want to, but I love that organising trips. Yeah. I want to get the tickets booked, know where I'm going to be at a certain time, I always approach things very casual, with the exception of where I'm eating. So I want to know where I'm eating on certain days, because I've got some really nice places lined up. And yeah, it's that's that. I'll be doing some bit more planning on that front, I'm sure.
No, well, I can't wait to hear about it. Of course, the Euros are going on. That's an exciting
the Euros, exactly.
Yeah,
I mean, I saw Scotland yesterday at least have a little bit of redemption from the first game, and then it's England this afternoon. So I'll be balancing work and football this afternoon. But
Scotland got buzzsawed by Germany, didn't they? They just.
I didn't see that because I was
5-1. Yeah, it's funny. I was watching it during dinner. It's on at nighttime, so it was on in the bars and stuff. And
but I did enjoy the game yesterday. Yeah. If the post was a little bit further to the left, they could have won it. But anyway, never mind. I think we should wrap up. And,
John, it's been a good show.
Absolutely, and we'll talk soon. Absolutely. Thanks a lot.